Hi friends, today we will be discussing about a research topic on nucleotide excision repair mechanism. Okay, now whenever this nucleotide excision repair mechanism is impaired in the organism, in the human body, then what happens? A disease called xeroderma pigmentosum occurs, and when a person suffers from xeroderma pigmentosum, these patients suffer from severe photosensitivity. They are very sensitive to UV radiation and high incidence of sunlight induced skin cancers. Okay, this nucleotide excision repair mechanism is there. It consists of a general pathway termed global genome repair, which remove the lesions from the entire genome, and a specialized pathway referred to as transcription coupled repair mechanism is there to repair these lesions which appear in the organism due to UV induced damage. And the name of this repair mechanism is nucleotide excision repair. Okay, now this transcription coupled repair, in short we will say as TCR repair, is confined to DNA lesions in the transcribed strand of transcriptionally active genes, and it strictly depends on ongoing transcription by RNA polymerase II. That means whenever there is transcription coupled repair, it is dependent on the is there in the system, and it helps in repairing the DNA lesions. Okay, here RNA polymerase II is involved. Now, whenever a person will have this defective transcriptional coupled repair mechanism, then in that person an inherited disorder develops known as Cockayne syndrome. So the person having defective transcription coupled repair, they have Cockayne syndrome, and this disease is inheritable. Okay, so now this you must be knowing that how the repair mechanism occurs in the organism, that incision of the damaged DNA, whenever the DNA is damaged by high photoradiation, then what happens? The damaged DNA has to be incised, it has to be cut from there. So this whole process of repair is a multi-step process which involves the recognition of the damage at first, followed by that region will be opened up because DNA helix is like the helical, this is a wounded one, so this will be opened up around the lesion. Then there will be dual lesion, means from the both the strands that the lesion will be removed, and subsequent excision of oligonucleotide containing the DNA lesion. The DNA lesions having the oligonucleotides will be removed. Okay, now it was explored by the scientists that which is the first protein which goes and binds to the DNA lesions specifically. So scientist was exploring a protein named XPC, small h capital H R23B. They found that whether this is the first protein which bounds to the DNA lesion or not. Their conclusion was that this protein is able to bind the normal DNA as well as the DNA which the lesion where the lesion is present. So it cannot be concluded that this is the protein which binds to the DNA lesion, since it is able to bind both the normal DNA and the DNA with lesion. Other scientists found that RPA protein and XPA protein, they are the initial damage sensing factors of human excision nuclease. Okay, so this assembly of these nuclear excision repair factors on the DNA requires the involvement of some transcription factors also. For example, transcription factor 2H and some structure specific endonucleases, example ERCC1, XPF, XPG. Here, the transcription factor 2, which is the basal transcription factor, it exerts a dual function in the cell. First of all, it is being an essential factor in RNA polymerase 2 transcription initiation and also in nuclear excision repair. Okay, if we go into the detailment of the structure of this TF2H, it consists of 6 polypeptides that are indispensable for both this NER, which is nucleotide excision repair, and transcription initiation. The two components of this core TF2H, which is XPB and XPD proteins, they are helicases which are responsible for unwinding the DNA helix in the nuclear excision repair. So remember, XPB and XPD, they are responsible for unwinding the DNA double helix where the nucleotide excision has occurred. Okay, has to occur, where the nucleotide excision has to occur. Now, once single-stranded DNA is generated as an intermediate, this is stabilized by RPA proteins. Now, the identity of the proteins which is responsible for binding of TF2H to the site of DNA damage and their sequence of action have not been fully clarified yet. It was suggested that TF2H protein, it interacts with XPC, which is the first one to bind to the lesion, bind to the damaged DNA. Okay, indicating that XPC attracts TF2H to the lesion. Now, for every finding, there has to be the analysis done in the lab. For here, the analysis done was number one, the nuclear distribution pattern of proteins, how they are distributed in the nucleus, it implicated in the damage recognition and in some incision steps of nucleotide excision repair. These proteins were assessed by immunofluorescent labeling using antibodies which were raised against XPA, XPC, XPB subunit of TF2H. Okay, and also antibodies against ERCC1 and XPG. In growing cells, fluorescent images of various proteins consisted of numerous small foci dispersed throughout the nucleus. So in growing cells, they all are being dispersed throughout the nucleus. Additionally, there is a protein named ERCC1. It exhibited larger and brighter focus throughout the nucleus, which were also present in living cells. And whenever they have to repair, there was cluster of this TF2H and ERCC1 was seen. So it can be said that due to UV light, when there is a change in the UV light, then the distribution pattern of transcription factor 2H changes. Now, I would like to discuss with you the nucleotide excision repair model, also known as global genome repair pathway. At first, the damaged lesion will be recognized by a protein named XPC HR23B. After this, there will be the recruitment of TF2H. I said that XPC is the one which is helping TF2H to recruit to the lesion. Then after that, there will be the helix unwinding. Then there will be XPG recruitment. After that, there will be XPA recruitment and RPA recruitment. This RPA will stabilize the single strand. After that, it will be ERCC1 and XPF recruitment. After that, the formation of incision complex is completed. So what is the sequence of recruitment? First is XPC, then G, then A, and then F. Okay, C, G, A, F. So that was the sequential assembly of the nucleotide excision repair factors in vivo. If this has helped you, kindly give the feedback. Thank you so much. Keep updated, keep subscribing, and stay tuned with me. Thank you. Hi everyone. Today the topic I've selected is human Werner syndrome. This syndrome is early onset of aging and it is due to elevated chromosomal instability, and a high incidence of cancer is there in this syndrome. The Werner protein responsible for this syndrome is a member of REC-Q gene family, but this REC-Q gene family is a very big family where Werner proteins are produced, but these Werner proteins are different from other REC-Q proteins in that these Werner have unique 3' to 5' exonuclease activity. Okay, The human Ku heterodimer, it interacts with Werner protein and stimulates Werner's exonuclease activity. The scientists have worked upon this modification of this Werner exonuclease activity by DNA-dependent protein kinase. It has been found that this DNA-dependent protein kinase, they do not alone affect the Werner exonuclease activity. In addition to that, Ku mediates a marked inhibition of it. So it was seen that DNA protein kinases, they inhibit this Werner protein, and here this kinase activity of this protein is required to inhibit that protein. So WRN, which is Werner protein, it is the target for this kinase phosphorylation, and once it is phosphorylated, and once for its phosphorylation, it requires the presence of Ku, which means that both Ku and DNA protein kinase are responsible for the inhibition of this Werner protein. So this Werner syndrome is a human autosomal recessive disorder, and this shows the characteristics like graying and loss of hair, wrinkling and ulceration of skin, atherosclerosis, osteoporosis, and cataracts. It also exhibit an increased incidence of diabetes mellitus, type 2, hypertension, and malignancies. Okay, the gene which is responsible for this Werner syndrome is WRN gene, and defects in which is responsible for this Werner syndrome. This protein WRN, it is 1432 amino acid protein. It has 3' to 5' helicase and a 3' to 5' exonuclease activity. It has an important role in DNA metabolism. The precise cellular roles of both the helicase and exonuclease activities remain to be undetermined. So, what is the result of this whole experiment? It was seen that this DNA protein kinase, it alone does not affect this exonuclease. This Ku can interact physically with this WRN protein, and it functionally stimulate WRN 3' to 5' exonuclease activity. So this Ku and DNA protein kinase, they are the subunit of DNA protein kinase. Okay, and they are involved in the repair of DNA. So it is said that this WRN exonuclease activity, it is inhibited by the activity of DNA protein kinases in the presence of Ku protein, not alone. So thank you so much guys for studying about the role of Ku protein and DNA protein kinase in inhibition of WRN protein, which is responsible for causing Werner syndrome in human cells. Thank you. Hi friends, today our topic of discussion is effective fluorescent sensing of lead ions and live cell imaging. So we all know that toxic heavy metals, they create a pollution and they are a serious hazardous for environmental health, and it has received growing attention in worldwide because their toxic effect is latent towards human health and animals. So lead is considered as one of the most hazardous heavy metal ions in the environment. It can cause problems such as brain damage, neurological, cardiovascular disorders, and mental retardation. Recently, some techniques have been employed for lead detection, such as atomic absorption spectrometry and inductively coupled plasma mass spectrometry. And these techniques require sophisticated and expensive instrumentation, which hinders their practical application for lead ion detection. Recently, fluorescence methods have also been enabled for the development of new detection process because of their more sensitive and faster detection of lead ions in aqueous solution. Fluorescence methods are extremely attractive because of its cost effectiveness, ease of operation, simplicity, and high sensitivity. In recent years, Some DNA enzyme, sensor, aptamer, polymer dots, and inorganic nanomaterials like gold, CDS, ZNS have all been developed to monitor and analyze the lead ions by the fluorescence method. Some scientists have also identified and developed liposomes and magnetic nanoparticles for lead ion sensing. Unfortunately, use of this DNA enzyme, aptamer, gold nanoparticles, they are limited for routine analysis due to their cost effectiveness. Application of inorganic quantum dots is also imperfect due to the effect of toxicity caused by heavy metals, while liposome and magnetic nanoparticles suffer from multi-step synthetic approach. Therefore, it is highly demanded to synthesize a simple and inexpensive material to apply as excellent selective and sensitive probe for lead ion detection. So fluorescent CD, they have been received and receiving increasing interest in research owing to their potential applications in the area ranging from imaging, sensing, and drug delivery to optoelectronic devices. Compared to the conventional organic fluorescent dyes and heavy metal based quantum dots, CDs, they have created great focus because of their facile synthesis, splendid water solubility, low toxicity, high photostability, low cost, and excellent biocompatibility. So, friends, CD is what? CD is carbon dots. They were effectively employed in the detection of heavy metal ions. So how are these carbon dots were synthesized? They were synthesized by hydrothermal method using Tulsi leaves as raw material. Since Tulsi leaves contain large number of organic molecules, say for example rosmarinic acid, olenolic acid, ursolic acid, carvacrol, and many more. These organic molecules are converted into carbon dots by hydrothermal treatment at 180 degrees centigrade for 4 hours. The synthesized carbon dot shows strong green emission under the UV light at 360 nanometer. These synthesized carbon dots were applied to the detection of trace amount of lead ions in real water samples, and can also be used for triple negative breast cancer cells imaging. The synthesized carbon dots were characterized by transmission electron microscope, X-ray crystallography, and UV visible spectrophotometer analysis. So what is the final result of this whole research paper? It was found that highly fluorescent carbon dots, they have been fabricated by one step hydrothermal method from natural source. Most importantly, no any additional chemical functionalization of carbon dot is required, which provide the potential opportunities of simplicity and cost efficiency. These carbon dots have good photostability and are being chosen as sensing probe for label-free sensitive detection of lead ions. The practical application of this probe for lead ion detection has been successfully demonstrated in real water samples. This fluorescent probes also show good cell permeability and low cytotoxicity, thus effective use has been there for fluorescence imaging of MDA MB468 cells. This is the first reported method to synthesize fluorescent carbon dots from Oxymum sanctum, that is Tulsi plant, in a single step and can be applied promisingly in bioimaging. Thank you so much for your attention. See you. Stay tuned. Bye.